So, Don, it is an absolute pleasure. I welcome Professor Don Layman. Don, I just to save time because there's a lot of stuff that I think we should cover. Professor Layman's arguably one of the world's leading authorities on protein metabolism, muscle protein synthesis throughout his career. He's been doing it for his whole life, and just to let people know who we're chatting with, you've got a lot of experience around protein. We've covered this before, but I think it bears repeating, and then there's some other stuff I want to maybe address if you don't mind. So let's talk about protein requirements because RDA 0.8 grams per kilogram. There's people that even suggest lower. There's people saying we need even much lower than that we can actually, because we are constantly re-ingesting sloughed off cells from our digestive tract and we're recycling mucus, that we might need 40, 50, 60 grams of protein a day for an average sized person. What are your thoughts on protein requirements and why or why not is that sensical or not?
A lot of complexity to that question. The origins of the RDA, which is defined as the minimum level of protein to keep 97% of the population from showing deficiency symptoms, is defined originally by nitrogen balance because it was defined way back in the 60s and 70s. And basically, it was done with young adults. And so that's really been the basis of it. In the last 20 years, we have slowly realized that as we get older, we become less efficient with protein. And we've developed other techniques like muscle metabolism, muscle protein synthesis as biomarkers beyond nitrogen balance. So I think most everybody agrees that for most adults, and frankly, I define adults, older adults as anyone over 35, we really need something above 1 gram per kg and probably 1.2 or higher, depending on what your goals are, muscle gain, body composition changes, or just staying healthy. So I think the RDA is what it is. It's designed to prevent a deficiency, and it kind of does that reasonably well. But that's different than saying what's optimum for health. And I think optimum for health is quite a bit higher.
Yeah, Don, this is something that I'd heard recently about the nitrogen balance study. So to explain how nitrogen balance was calculated and why do we think that's inadequate for informing us on how much protein we need?
So nitrogen balance is basically determining all of the nitrogen that's going into your body, and it's not determining amino acids, it's determining total nitrogen, which could be from other kinds of things like nucleic acids. And then it's trying to measure all of the outputs, everything from urine and feces, but skin and nitrogen from your breath, and trying to capture all of those. And unfortunately doing it in a fairly short time frame. So the way it was always done was basically taking people down to very low levels and asking where could they come into balance. And usually in a 7-day period, people would come into balance somewhere around that 0.8 grams per kg. But again, that's totally different than asking what's optimum for health. It just means that you're not steadily losing huge amounts of nitrogen, which is a pretty crude measure.
Yeah, there's a fellow named Peter Ballerstedt who talks about crude nitrogen or crude protein, and it doesn't reflect amino acids, or particularly essential amino acids. It's just the nitrogen in foods, and we see that a lot of times when we compare protein on the grocery store label, particularly with kind of plant-based foods. Is there any thought behind what's usable versus what's not when it comes to protein on a food label?
Yeah, I mean, protein is complex because we have 20 amino acids that we have all the time. One of the lines I like to use a lot is saying we have a protein requirement is like saying we have a requirement for a vitamin pill. We don't actually need the pill, we need the 12 vitamins that are in it. And for protein, we don't actually need protein, we need the 9 essential amino acids that are in it. And particularly lysine, methionine, tryptophan, and leucine are always limiting in plant-based proteins. And so how do you balance those correctly? So we really ought to be talking not about a protein requirement, but we ought to be talking about a lysine requirement, and a leucine requirement, and a tryptophan requirement. We talk about B12 and B6 and vitamin C. We don't talk about a vitamin pill requirement.
Yeah, and I know you authored a paper talking about I think the role of red meat or something along those lines a few years ago, and one of the points you made out was, if we switched over to a plant-based diet, we would have some critical deficiencies, and I think one of the nutrients you point out was lysine. Can you talk a little bit more about that as far as how important is lysine? Why is it so important? And what would happen if we, what would change with the calorie requirements to get lysine if we opted to go with a meat-free diet?
Yeah, so as you look at plants, certain kinds of plants are more limiting in individual amino acids. So grains in general, wheat and oat, corn, are all very limiting in lysine. Legumes like soy and pea are limiting in methionine, the sulfur amino acids. And we can go on, look at tryptophan and leucine. So the problem is most Americans get the majority of their plant-based protein from grains at this point. So it's very limiting. And it's absolutely true you can get all of the essential amino acids from plants, but you will have to eat more total protein and typically more total calories to get to it. And the problem with most vegetarian type diets is not only do you switch to poor quality protein, lower bioavailability, but you also reduce the total amount. So you've got all three things going against you at the same time. So it's not that it can't be done, but it takes a lot of food skills to pull it off and do it well.
All right, one other topic, and again, I know you and I have covered this before, but there is still a lot of talk about mTOR being leading to cancer and premature aging. And one of the studies that I know you've had a criticism on was first of all, there's not a lot of human data on there, and the only real human data study that I've seen, unless you've seen others at this point, was one that Levine and Longo put out in Cell Metabolism a few years ago, and I know you guys question some of the methodology and the interpretation and the results. Could you discuss about mTOR? Is mTOR universally bad? Should we be avoiding protein to prevent cancer and premature death based on the work of Longo, Levine? And maybe you can reiterate what was the problem with that study.
It's been a while, so I can't really go back into the details. I hadn't looked at that for a couple of years now, so to actually critique the details. But the basic problem with that is that they have an agenda against animal proteins. And so they have been willing to distort data to basically achieve their agenda. The problem with how people have run those studies is they've based a lot of it on animal studies. And what they do is they take basically a mouse and they ad libitum feed them and then look at different protein levels. The problem with ad libitum feeding is that you now have what's really a chronic stimulation of mTOR. And if you think about protein is consumed in meals. If you look at mTOR signaling, mTOR is stimulated by a number of things. One of them is protein, leucine, but it's also stimulated by insulin, IGF-1, energy, carbohydrates in particular, and some other things. And so the problem with it is they're ignoring the fact that the animals are overeating too many calories. They're eating chronically 24 hours a day. They have too much insulin. And so they chose to blame protein when in fact it's always an insulin carbohydrate problem. And the issue with protein is we need to eat it in pulses. We want mTOR to trigger, and then we want it to go into an idle phase. The worst thing you can do is graze all day long and keep mTOR active all day long. Anyway, so mTOR has lots of different roles in every different tissue, but in muscle it has a very unique role in terms of, especially in adults, in terms of how we maintain our muscle mass. And it's critical to understand how to keep that balance correctly.
This is something great. This gives me a good segue into something I wanted to ask you about. So let's look just like protein absorption kinetics, and A lot of the studies that are out there are done on whey protein, which is a very rapidly absorbed protein. It's because it's so small and it doesn't take much to digest it. When we look at like a mixed meal, like for instance, I had 1.5 pounds of meat for breakfast this morning, and then I went out and lifted some weights, and I did some sprints, and I went for a walk, and now I'm here. So that's what I did immediately before I hit here. So that steak that I had, an hour and a half ago, and I actually worked out pretty much right after because I usually can. But what's going on? How long is it taking for those amino acids to get through the digestive tract, through my stomach, gastric emptying time, to be absorbed into my bloodstream? And then how long are my muscles going to be receptive? When do I see a peak amino acid levels? When is my muscle sensitivity towards absorbing those? What's going on with all that stuff?
I honestly have never seen a study where somebody actually evaluated 1.5 pounds of meat at a time. So I can't specifically speak to that. We know that protein and fat will slow down gastric emptying. So the length of absorption, the more you eat, chances are the longer it will still be an absorptive pattern. What has been studied is a lot of individual proteins in sort of bolus meals. So like you talked about whey protein or soy or wheat or casein or things like that. And we know that in a pure sense like that, most of those proteins will be digested and absorbed in about an hour. What's interesting in amino acids, though, is while the meal is digested and absorbed, amino acids will pool in the blood and in the free pools for up to like 5 hours. And so after a meal, you'll have elevated blood levels for a fairly extensive period of time. What's interesting within that is that muscle protein synthesis will only last about 2 to 2.5 hours. So while your meal is still being absorbed, and you still have high blood levels, you're actually not getting a lot of benefit to it. So while you're consuming a lot of your calories with the meat for lots of different reasons, as far as a muscle effect, you're way over consuming the amount of protein that you're getting a muscle benefit from. I mean, there's calorie balance and all kinds of things that you can choose to do it for, but one of the things I like to talk about is that there's a range of meal response. And depending on the exact protein you're using, that's probably somewhere between 25 with a really high absorbing protein like whey, up to maybe 55 grams of protein. Beyond that, you're probably not getting much of a muscle effect. So you may get whole body effects. You may have a larger liver or a larger kidney or a larger GI tract, but you can't make muscles larger just by having larger meals.
Yeah, so I think you would say more, maybe perhaps optimally smaller meals more frequently, if because
there's a huge problem I think in the way people are talking about meal distribution, and it's a combination of two things. One is this response range of a meal, and then the next is a leucine threshold concept in terms of a meal, and then the distribution. One of the things that has been misinterpreted from the paper that Doug Patton-Jones and I published, I think in 2014, is that you need an even distribution of protein through the day. And that's totally false. The issue is what you need is how many meals per day exceed the leucine threshold. How many times do you trigger that anabolic threshold? And the two meals that are most critical are your first meal after an overnight fast and probably your last meal, a dinner meal, because they're farthest apart. No one, to my knowledge, has ever shown a leucine response to a lunch meal. So basically, I look at meal distribution as I want to have a first meal, and then the response range I talked about a little bit ago was 25 to 55 grams, somewhere in that range. So for me personally, I want that first meal to be in the upper part of that range. So I'm having 45, 50 grams of protein, not quite 1.5 pounds of meat, but I'm having a fairly high protein meal, and I have the last one. The middle meal, I'm kind of in weight maintenance. I'm not really looking to gain muscle mass. I kind of don't pay that much attention to my noon meal. And, other meals, I look at meal sequences. If you're kind of an adult, happy with your body composition and weight, 2 meals a day is perfectly fine for protein. If you're in a catabolic condition like weight loss, or you're trying to change your body composition, I think 3 meals a day is a better choice because you have more anabolic periods. And if you're in an anabolic period like you want to build muscle, then I think 4 periods per day. But you get only about 2.5 hours per meal. And so how many of those do you want per day?
Yeah, that's great information. Let me ask you about so muscle protein synthesis. Now, in talking with you and Stu Phillips and some others, I mean, when I hear what are the essential requirements for it, it seems like there's a leucine threshold. It's having a balance of the remaining essential amino acids in that composition. And then it is, a minimal threshold for insulin, which can be permissive, and then a caloric surplus, and then obviously the resistance training is going to be a stimulus as well. Is that pretty much it for what's required to build muscle, or is there some other things that we have to consider?
Obviously, the resistance. I mean, first and foremost, if you're going to build muscle, you have to have the resistance training. So you have to have the dynamic need to increase your myofibers content. As far as the nutritional support for it, I think you captured it exactly right. I mean, it's, you have to think about these leucine thresholds. You have to stimulate mTOR. We know that muscle protein synthesis is always running, maybe 35, 40%. But what leucine does is trigger the extra capacity. It triggers certain mRNAs to express, and it's the ones that actually increase your capacity to build myofibrillar proteins. So it's targeting certain mRNAs. So it's not like you're going from 0 to something. You're going from 40% to 100% max is what's happening. And that can be sustained for about 2.5 hours. Why? We don't really know. Phil Etherton called it a muscle full concept. My research group suggested it was an energy dependent aspect, that muscle protein turnover is just so expensive, it can only run so long and you run out of ATP. We don't really know why, but there's pretty much agreement that a meal will only run so long. And what's interesting about that is the mTOR signal is still probably active 5 hours later, but protein synthesis has pretty much shut back down after 2.5 hours. And that's one of the reasons I say no one's ever really shown that a lunch meal is important, because mTOR is already active. I think a lunch meal with 15 or 20 grams of new protein might be perfectly adequate, because you don't need to stimulate mTOR, it's already stimulated.
Yeah, and some people just skip lunch altogether, probably without too much.
I actually do. As I've gotten older, one of the things I realize is I need a lot less calories. And so I try actually to compress my 2 meals. So my first meal frequently doesn't happen till 10 in the morning or something. And my last meal might be 5:30. I try and compress those eating periods into a narrower range.
Yeah, and so if we have the essentials, protein, obviously, muscle is made out of protein, and that makes sense in saying protein is what you need to put on muscle for the most part. What role do the other macronutrients have outside of caloric surplus, either carbohydrates or fat? And do they have a differential role on muscle protein synthesis that you're aware of?
Not particularly. I mean, you mentioned that insulin in adults. I mean, insulin in children is a growth hormone. As long as you're in a growth phase, growth hormone, IGF-1, insulin, those are driving growth. Once you get out of growth, once you get past your mid-20s, now it's diet quality. So insulin takes on a passive role. Having some carbohydrate with the meals probably okay, but you'll actually get enough of an insulin response just with the branched chain amino acids. So you can get an anabolic response pretty much just with protein, as long as you have enough calories in the meal to also make it, an anabolic period. It's hard, it's hard to gain weight. It's hard to gain muscle mass in a negative calorie balance condition. It maybe can be done, but kind of the best you can do is sort of maintain status quo. Yeah,
I've seen people talking about gaining weight while in a deficit or gaining muscle in a deficit, and I think that that's pretty unusual, and maybe there's some drugs that come into play with that.
And I don't think they can sustain it very long. I mean, you might be able to do it for a couple of weeks or a month, but you're not going to sustain that with, in a 4-month period.
Let me ask you about Gluconeogene Genesis, Don. Do we know, I mean, when I go back and read some of the old literature, 1920s, 1930s, the presumption is somewhere around 55% of protein or amino acids can be converted into glucose theoretically, but then if you look at the isotopic data, it never even comes close. What is the actual percentage of protein that's available for gluconeogene? And I realize it may depend upon the state of the liver and the glycogen status, but what? And the other thing is, how rapidly does it occur? Because this is a thing that I see, because I see this in diabetics, and I think it's more has to do with glucagon than gluconeogenesis, and I think it's due to glycogenolysis because of glucagon stimulation on the liver releasing glycogen more than gluconeogenesis. What is the time frame of gluconeogenesis? And then what is how much of that protein can we convert into glycogen or blood sugar under different circumstances?
I don't know that I know any exact percentages. I mean, your original comment, if you just look at the carbon skeletons, about 60% of protein is going to be metabolized by glucose mechanisms. As you oxidize them, you have to oxidize. Every amino acid that you eat ends up getting oxidized in some way. And 60% of them are going to get oxidized through carbohydrate type metabolism. Does that actually produce blood glucose? It kind of depends on what tissue it's happening in. In terms of your other statement about time frame, one of the great things about protein and gluconeogenesis is it's very slow compared to the effect of eating carbohydrates. I mean, carbohydrates, when you eat a meal that contains carbohydrates, that will enter the blood and has to be cleared in 2 hours, or that's the definition of diabetes. If you're not back to baseline within 2 hours, that's diagnostic for diabetes. It's a toxic substance. Amino acids, they will pool, as I said earlier, in the blood for at least 5 or more hours. And so you're going to be slowly converting that carbon into glucose or fatty acids, ketogenic, over at least a 5-6 hour period. And so it's a very slow process and very continuous. One of the things that we always saw in a lot of our animal studies is one of the cool things in when you have a high protein, low carbohydrate diet, you actually maintain higher blood glucose in the middle of the night. You don't go into those hypoglycemic states where you wake up starving because you're actually using amino acids to continuously make glucose. And the body sort of adapts to that. Gluconeogenesis ranges normally from a low of maybe 2 grams per hour to a high of about 7 grams per hour. You can see numbers really high like 10, but those are acute studies that can't be maintained. So there's a range. So you never totally shut it off, and you can activate it to a certain level. Under normal circumstances for the average American who has a high carbohydrate diet, the vast majority of gluconeogenesis is coming from recycling of lactate. It's glucose being recycled. And the next part is, amino acids, but actually alanine is being recycled. Glucose is also probably the predominant gluconeogenic substrate for most Americans.
Yeah, I'd heard in addition to alanine, I read a study looked at, I think it was either glutamine or glutamate being a very, very gluconeogenic substance as well. So let me just go to that statement about the nighttime lack of hypoglycemic episodes, because a lot of people, particularly diabetics, wake up with this thing called a Somogyi effect or dawn effect, where they have this glucagon cortisol awakening effect, and they have very high blood glucose. Do you think is there any evidence that elevation of glucose in the night would blunt that effect in the morning? That is to say, your body might say, hey, we don't need as much of a dawn effect because we're already, I don't know if there's any research on that, or I know. I
can't address the specific question, but what we do know is that with metabolic syndrome, with type 2 diabetes, that higher protein, lower carbohydrate does stabilize all of those hormone swings. What you stop getting are these big rapid swings. People tend to need a lot less drugs, metformin, insulin, etc. So it definitely stabilizes it. People on higher protein, lower carbohydrate diets tend to run slightly higher cortisol all the time, which I think is sort of indicative of the fact that the gluconeogene genesis is running a little higher all the time. I see no problem with that. My feeling about the glucagon story is glucagon's a very noisy hormone. I think the real swings are insulin swinging up and down around glucagon. Is insulin high? Is it low? Does it, if insulin's high, glucagon becomes irrelevant. If insulin's low, glucagon becomes more important. But glucagon kind of stays more stable, and insulin swings wildly.
Yeah, I guess the caveat to that would be if you're very insulin resistant, then the insulin becomes less relevant and the glucagon probably becomes more relevant. Let me ask you, this is another shift. I see some thought in the literature. There's this concept of, well, it's not a concept, it's a fact, that there's something called branched chain aminoacidemia, and it's been associated with metabolic syndrome, insulin resistance. What are your thoughts around that? I've talked to Ben Bickman, who thinks that's reverse causality that we see with diabetics. They tend to be in a catabolic state, and therefore there's more of those amino acids running around. But what are your thoughts on that sort of thought, that eating too much protein is going to cause branched chain aminoacidemia, and therefore it's going to lead to metabolic syndrome, or insulin resistance, rather?
I think it's total nonsense. The thing to always remember is that glucose is a highly toxic substance in the body. It's an interesting thing in that it's an essential fuel for brain, neurons, red blood cells, but it's also one of the most toxic substances the body experiences. And so it's very tightly controlled. But if you have elevated blood glucose, it shuts down metabolism of everything else. First and foremost, you have to get glucose under check. And when you do that, you stop fatty acid oxidation and you stop branched chain amino acid oxidation. And so you'll get a buildup of free fatty acids and branched chain amino acids in the blood, but it's secondary to the excess glucose. You bring glucose intake back down and you'll correct both of those other issues.
Somebody was asking about, is there any kind of diurnal effects on, when's the best time to train? Is that data out there? I mean, I've seen some stuff where people set PRs in the afternoon in athletic training, but I haven't really seen a lot as to when is the best time to train for, I guess it depends on what your goals are, but is there much that you can talk about on that?
I am definitely not an expert. So my very strong opinion is whenever it makes you feel good. Whenever you get off your butt and go do it, that's the best time.
Yeah, and I think that's a pretty reasonable.
I mean, I personally I like working in the morning at the computer, then I get bored of it, and like 2 or 3 in the afternoon, I want to go out and exercise someplace. So that's my style. The last thing I want to do is roll out of bed and go exercise in the morning. So that's my style, and I'm 70 years old and I have the same weight as I graduated from high school. So it works for me.
Yeah, it just makes sense. If it works, it works. Somebody was asking about fasted versus fed training, and can you define? I mean, how do we define fed training? I mean, what's the time frame of that? Are you fed training if you ate 6 hours earlier or 3 hours earlier? Thoughts on the differences between those two?
The original arguments for that I saw were that, well, if you train fasted, you'll burn more fat, but that's ignoring that it's a calorie balance issue. I mean, basically, you're going to burn whatever calories in a 24-hour period that your body burns, and whether you burn them right after exercise or just before you go to bed, it doesn't really matter. So I don't really buy that. I think, again, it's really the issue is how do you feel? Some people perform better that way. Some people don't. You commented, you ate 1.5 pounds of meat and then went out and exercised. That would make me ill. In terms of protein relative to meals, I think there's far more evidence that protein after meals is more beneficial than protein before meals. I still have never really been convinced that protein directly ahead has any real beneficial effect. So, but I'm pretty convinced that protein after does, certainly in early training phases.
Is that, and again, I would just question on that statement, is that basically based on whey protein studies with the quicker absorption profiles with after meal, or do you know?
I've seen it done with meals, but I think you're right. I think the majority of the pre and post have been done with free proteins, isolated proteins. And, the easiest one to study has been whey, just because it's easy to mix and study. So, yeah, I think you're right in that.
Yeah, like I said, I'm just wondering if I've got a ribeye steak in my belly and it takes 3 hours to get through the digestive tract, and then, I train 2 hours later, and then an hour later it might be hitting my bloodstream. So I'm just wondering,
Yeah, I've never seen any data to support or reject that concept at all.
Yeah, I guess the studies just aren't done. I guess it's too hard to do studies with ribeye steaks and then whey protein, probably too costly.
Well, and the question is, how do you fund that study? Who's going to pay for that study?
Yeah, sure. Yeah. Well, I don't know how many people you'd need. You might be able to do it in a small sample size, get everybody steaks and measure their blood amino acid levels, after eating a steak.
I mean, absolutely, you're going to have higher blood amino acid levels during it. The data that supplying free amino acids to people during endurance exercise is pretty weak. So, what's the benefit of having higher amino acid? I don't know. The kind of an interesting thing is the way we actually discovered leucine and the branch chain amino acids was actually, we were doing a carbohydrate study. We were actually doing it for Gatorade. And Quaker Oats, and we were studying different pre-exercise cereals in elite cyclists. And what we found is at the end of these elite cycle power studies was that the branch chain amino acid in the blood dropped like a rock. And we thought, what in the world does that do? And so we then looked at protein synthesis and saw it fell off after these exhaustive exercises. And that's where the whole concept of leucine supplementation and Mtor and EIF4 came from. We started working with Jim Jefferson and Tracy Anthony and Josh Anthony and and that's where that whole concept came from.
Interesting. Somebody was wanting to comment on taurine. Do you know of any unique or special concerns around taurine with regard to health or muscle protein synthesis?
There's been some recent people trying to make something out of that. Taurine in general is it's a sulfur amino acid metabolite. It comes from cysteine. And people have been trying to make some comments about cystathionine beta synthetase and genetic flaws and all sorts of things like that. Taurine and one of the other metabolites, glutathione, tend to be low in the American food supply because we're fairly low in cysteine. I think there's a story that we don't yet understand about methionine-cysteine ratios. I keep sort of thinking about that one. But in general, taurine, carnosine, glutathione are all fairly low in the American food supply, and that's because our cysteine levels are on the low side.
Yeah, I was going to ask about, let me see, I think someone else had a question in here. Let me get into this because this is another thing that I think it bears repeating. A lot of belief out there on kidney function and protein. Protein harms our kidneys is what we continue to hear, and a lot of physicians continue to sort of say, what is the data on protein and harm for kidneys show actually?
Yeah, there have been great reviews. There's one by Stu Phillips. There's been 2 or 3 in the last 3 years, and they overwhelmingly show that protein does not cause kidney problems. In fact, all of the direct studies show that kidney actually enhances renal function. It enhances the glomerular filtration rate, the rate of clearance. It makes the kidney more efficient. It's kind of like saying, you shouldn't exercise because it makes your heart work harder. Protein makes your kidney more functional. the whole origins of that have been renal failure. And particularly diabetic renal failure. We know that when the kidney actually is failing, end-stage renal disease, that you have to eliminate solutes, things that the kidney has to clear. So salts, for example, have to be reduced, but so does urea, so does protein. So basically, we have to reduce the amount of things that the kidney experiences. But there's a debate in the nephrology field as to when that should occur. Should it occur at stage 4 renal failure, stage 3, stage 2? Years ago, people thought, well, you immediately need to do it. But now people realize as soon as you lower the protein level in the diet, the kidney begins to shrink in size and you accelerate the deterioration. So most nephrologists aren't actually reducing it until they get into stage 3, even stage 4 renal failure. So that is a myth that continues to be perpetuated out there that has no basis. Even back in the 2002 range, when the National Academy of Sciences was establishing the DRIs, they reviewed that very topic and said, in fact, the only relationship to protein to renal function was the more protein, the better the kidney functioned.
Yeah, and I've seen, just within these folks that go on higher protein diets in this community, see improvements in renal function. GFR improves. One of the caveats of that is, sometimes we'll see on a higher protein diet, creatine, serum creatinine rather will rise, and that is how the GFR is calculated. And so I often tell people to get a cystatin C, and I'm not sure if you're familiar with that particular test, but it excludes protein as a confounder. One of the questions somebody had was regarding, you see this sometimes, bodybuilders talk about this, and they are often on high protein diet, and then some of the early research on creatine was associated with cramping. Do you have any thoughts on dehydration and protein, and do we need to take in more fluid when we do that, or are there any relationships between that and muscle cramps?
I mean, dehydration is a huge issue, and especially for endurance, athletes type of thing, and definitely relates to cramping. I can't really address the creatine level. I mean, obviously, creatine is something you take in fairly high amounts. I mean, it's, some people may be taking it in 10-gram levels. So that's a solute. Protein in general requires more fluid. One of the things we always saw in our weight loss studies, particularly with women, is we would take them from fairly low protein diets to higher, and they would often experience constipation in the first week or so. And that's really a dehydration issue. The protein just is hyperosmotic. It will draw water to it. And you just have to be conscious that you're going to need more fluids to go with it. Carbohydrates, generate more water when they're metabolized than protein or fat does.
Let me talk about liver function, because one of the concerns, in my experience, in a healthy human with a healthy liver, protein doesn't seem to be an issue. I think sometimes in cirrhotics where they have ammonia accumulation, we get these people with, they get toxic levels of ammonia and they don't do very good at converting it to urea so they can excrete it. What are your thoughts around protein and liver function in general? And then are there isolated instances where maybe it's a problem?
I mean, kidney, liver, GI tract are all very efficient in handling protein, but what you have to realize is it might require an adaptation period. So if you go from somebody who's consuming 60 or 70 grams per day and you want to get them up to 150, you should do that over a week to 2 week period, because all of those enzymes, all of those things have to adapt. So if you just take a person who's eating 60 and you give them 150 grams, there's a good chance you're going to see some ammonia in the blood, or you're going to see some diarrhea. You're going to see problems because it just takes the body time to adapt all of those digestive enzymes and the liver enzymes for the urea cycle and the kidney extract. All of those things have to adapt to the new normal.
Yeah, I think that's a great good advice, because I hear people talking to me, they say, yeah, I'm smelling ammonia in my sweat. And I say, well, it's probably from protein, like I said, probably is. Well, there clearly is an adaptation phase, and it may take a little. Some people may take a little longer than others. I think it just varies based on that. How much? So, would you say that? Well, let me go back to the caloric surplus for muscle building. How much of a caloric surplus do we need to be in? Do we just need to be in just a little bit, or does it have to be a massive surplus to build a muscle? I mean, what's the and how do? And then the other thing is, I guess, you know, because we talk about calories in and calories out, and I always have this caveat: as long as protein is equivalent. Why is that statement in there about protein being equivalent? Does it have a unique metabolic advantage as a macronutrient versus carbohydrates or fat?
I'm not sure I understood the second part. Equivalent to what? In calories?
Well, exactly. So some people will say that, you know, well, there's a thermic effect of protein, and therefore you could, you know, it makes a difference on the how much you can eat.
Absolutely. So yeah, in terms of a caloric surplus, you know, I think the answer to that is in what rate are you expecting to see a change? You know, if you're going to think you're going to gain 10 pounds in a month, then you're going to have a pretty heavy caloric surplus. If you just want to be a healthy adult and have more muscle mass at the end of the year than now, you just need to sort of be on a slight positive. So again, I think the answer to that is in what time frame do you think you're going to get a change? I think you just really need to be in a positive. It doesn't have to be massive. Again, unless you're trying to gain a whole bunch of mass in a short period of time. The equivalence is absolutely clear. I mean, the thermic effect of protein is somewhere around 15 to 20% of calories will be wasted as energy, where with carbohydrate and fat, it's around 5%. So there's a real difference in a thermic effect, and not to even mention the satiety effect is higher than carbohydrate. So from a calorie control standpoint, substituting protein for carbohydrate is always a huge benefit in terms of calorie control. A lot of people argue, you know, if you go back to freshman nutrition books and look at the thermic effect of protein, it'll always say, well, it's because of the digestion, absorption, and metabolism, which kind of means they have no clue what the cause is. I personally think that it's actually this muscle protein synthesis effect. That is a very energy expensive process. You know, we've measured ATP use. We know it's energy expensive. And I think that's why meal effect. So I think that having all of your protein in a single large meal will have less thermic effect than having it in multiple meals that reach these thresholds. So again, you know, I think distribution is important, but we have to understand that the distribution isn't based just on making it distributed evenly. It's based on how many times are you reaching these leucine thresholds of 3 grams or more. I mean, that's the key.
Yeah, I think does alanine have a role in there as well? I thought I saw something that alanine has like a secondary role behind leucine as far as, or maybe it was arginine. I can't remember, one of the two. But
I'm not really sure. I would probably make an argument for arginine, but I'm not sure I would for alanine.
Yeah, it might have been arginine. I can't remember. Sometimes they run together for me.
All sound alike, right?
Yeah, they all start with A. That's good. They're the same thing. No, I'm kidding. Someone was asking about protein or refractory period on protein distribution. I'm not sure I even understand that question. Does that trigger anything for you?
That's sort of what I was talking about how long a meal effect runs, so that basically, you know, what we found and others is that if you give a protein meal, you'll run muscle protein synthesis for about 2.5 hours. And then it's kind of refractory for stimulation. If just giving more protein on top of it doesn't really seem to make much difference. And so we always recommend that for the efficiency, you should space your protein meals at least 4 to 5 hours apart, because that allows for all of the mechanisms to reset, the ATP to come back into balance, and you can sort of redo it again. Refractory period, muscle full period, it gets different names, but the bottom line is muscle protein synthesis seems to have a finite period it can run, and then it needs to rest for a while.
And that's within the 24-hour period. Is there other evidence to show that that needs to happen on a more macro level, like, you know, people talk about training and then building in rest weeks and stuff like that? Is there great evidence that supports that, or is that something you've looked at?
Not something I've really looked at. I can't really offer anything to that thinking. I think that probably is a training concept of how long you can sustain really heavy training more than it is a metabolic concept.
Yeah, I mean, I pretty much train every day.
I don't really change my training much. As I've gotten older, I find that I have to sort of take a couple of days off during the middle of the week where I don't train heavy because my knees don't like it as much anymore.
What Brooke is asking about, does it make sense just to supplement, you know, 1 or 2 amino acids for muscle protein synthesis? I guess, you know, obviously leucine you talk about, but I guess that's assuming you're deficient in your food. But if you say you can't get it in your meal for whatever, how would you recommend supplementation for amino acids?
I mean, one of the things that we did, and it's been done by others, such as Doug Patton-Jones, is that, for example, we know that mTOR requires around 3 grams to stimulate it, maybe a little more than that, but somewhere around 3 grams to really stimulate mTOR, 3 grams of leucine. And so if, for example, you're having a small meal, like a lunch meal that only has 15 grams of protein and might only have 1.5 grams of leucine, if you put in another 1.5 grams of leucine, you can make that meal act like it's 30 grams of protein. So supplementing 1 in a small meal, and in that case, I would always supplement all 3 branch chains. You should probably never just supplement leucine alone. You should probably always supplement all 3 of them, because they metabolize together. So I can see that. I think we're definitely going to see supplementations of protein deficiencies. So, you know, putting lysine into a grain or something like that, I think is going to be possible. Methionine is a tough one because the sulfur amino acids carry a lot of taste profiles to them. So that's not very useful. But yeah, I think that supplementing some free amino acids can have benefits, but in general, I think you should eat the whole protein. I think it's just, I'm definitely a natural food person.
Yeah, I kind of think humans were designed to eat natural foods, quite honestly. Let me ask you this.
Free amino acids are so expensive.
Yeah, let me ask you this question. I hear a lot of people talking about say ketogenic diets being muscle sparing or protein sparing. Can you comment on the validity of that statement, or do you have any knowledge around that?
I don't have any reason to believe that. I think that muscle sparing is a protein-based thing. So, you know, a lot of the keto diets are fairly low in protein. So, you know, I think you need to be up in the 1.6 grams per kg of protein, and that's kind of high for a lot of the keto diets. So I've not seen any particular data that makes me believe that keto diets are more sparing than, you know, a more protein-centric thinking about protein sparing. So, yeah.
Somebody was asking about this is another concept that comes up. Acidity. You know, eating a high protein diet is leading to, you know, acidic urine, and, you know, I don't know. They talk about serum acidity or sulfur containing amino acids. Can you talk about what we know about those things? Does protein, is it acidic, and does it harm us?
So the metabolism of the sulfur amino acids does have an acidic nature to it. The original concept of that came out of calcium balances. And so on higher protein diets, you tend to see more urinary calcium. And so people jump to the idea that the acidity was causing bone loss. But when people like Kerstetter and others looked at it, what they found was that protein greatly increases calcium absorption. So you're going from calcium absorption of around 20% of dietary calcium to over 40%. And so you just have to excrete more in the urine because you've got more than you can use. The issue in terms of blood acid base is frankly silly in that the pH of the blood varies between, like 0.72 and 0.73. And if you get out of that range, you die. So obviously blood pH isn't changing. In terms of total body balance, most people now agree that vegetable consumption is the basic part of the diet. And the reason there would be a change is people aren't eating enough vegetables. So we don't have enough of the alkali part. It's not the acidic part of protein, it's the alkali part of plants.
Yeah, that's good information about calcium. I think that's a lot of people are calcium the osteopenia is a big problem, particularly in the aged. What is your thought on protein and bone health? Or do you have any thought on that?
I mean, all of the data from any of the fracture data, any of the sort of acute bone changes all show that higher protein diets are beneficial. What people sort of mistake is they sort of think, well, bone is a mineral thing. Well, bone is really a protein thing. It's a protein matrix that has to be formed first. And then what we do is deposit minerals on top of that matrix to give it structure or give it rigidity. But protein or muscle, bone first and foremost is a protein structure. And just like muscle protein turnover, bone has a protein turnover, and we have to support it. So I think sarcopenia and osteoporosis are the same disease. They're basically lack of resistance exercise and lack of protein. They're not lack of calcium. The United States probably has more calcium intake than almost any place else in the world, and yet we have more osteoporosis. It's not a calcium issue, it's a protein issue and exercise.
Yeah, I wholeheartely concur on that. I think our efforts to eat more calcium or supplement calcium have not panned out as far as osteoporotic fractures. And I think we woefully undereat protein, particularly as we get older, or we can't absorb it, as you alluded to earlier. We don't.
Yeah, I saw some data just last week about of young women in the United States with all this plant-based discussion that 20% of women in the 16 to 26 range are below the EAR. They're below 0.66 grams per kg. So those are frank deficiency symptoms. 20% of women in the United States. I mean, that's kind of a frightening number.
Yeah, especially since we're not living in a third world country where we have access to this stuff fairly cheaply.
Yeah.
It is kind of amazing. Don, is there anything else? We usually go about an hour and we're coming up on that. Is there any other topic you think we need to touch on that you think people need to know about?
No, I think we've touched on a lot of that. I definitely wanted to highlight sort of that concept of meal distribution, and it's not about, I've been meaning to write something and post it on Twitter. It's not about an even distribution at all. It's about how many meals per day do you want to hit these leucine thresholds. And I personally like a rather uneven distribution. I personally try and get 45 grams in breakfast. My lunch might be 20. My dinner might be 65. So I'm up and down. I like to push the upper limit of protein at a meal, and I like to have a high level in my first meal, and I kind of like having a meal that's a little lower. I like it being variable, but I'm getting at least 2 meals per day that are greatly exceeding that leucine threshold. So, I think we need to be more concerned about what's the optimal range to hit, and then being sure we hit that leucine threshold often enough.
Yeah, and again, I've seen you talk about 1.2 to 1.6 grams per kilogram being probably a solid range for most people, and then we've got some strength athletes and weight trainers that'll go up above that, and I've seen 2.2 or even higher. Does the data support that for those guys?
Yeah, no, that's exactly right. So for most adults, I would like to see it in the 1.2 to 1.6. For endurance athletes, sort of in that more 1.6 kind of range, and strength athletes, 1.6 to 2.2. I think those are totally normal ranges for those kinds of individuals.
Perfect. Well, this has been great, Don. It's always a pleasure talking to you. Where can people find you if they want to? I know you do a little bit of social media. It's good to see, getting the information. We need everybody in the world, everybody that's got a voice that wants to share this information. This is important information to get out there. So where can people find you, follow up on stuff they want to learn more about what you're talking about?
On Twitter, it's at Don Layman. I'm sort of reluctant to wade into Twitter very often. I find it just sort of blows my mind too quickly. And also my university email is simply Don Layman. I mean, dlayman@illinois.edu. So those are two ways people can reach me.
Yeah, I would say, the social media stuff can be a bit of a cesspool.
Yeah.
It's nice to be able to, the nice thing about these interviews, you can get some nuance out there. And I think that there's a lot of that.
I try to do it where I see some question floating around that I think I can add some value to it, but I tend not to respond to a lot of comments.
Yeah. Anyway. Well, Don, have a great day. It's been a pleasure. This will go out in a couple of days, I think, to the general audience, and I'm sure it'll be very well listened to, and it'll provide great information. Thanks again, Don.
Thanks, Sean. Always a pleasure.
All right. We'll talk to everybody later. Bye-bye now.